Hello, everyone. This is Pora Kim. I'm assistant professor in bioinformatics in the McWilliams School of Biomedical Informatics at the University of Texas Health Science Center, Houston. Today, I want to share my one of the fusion gene study in biomedical informatics. The title is FusionNeoAntigen, a resource of fusion gene-specific neoantigens. So as you know, there are different lengths, different size of mutations in cancer. The largest one is structural variants. The structural variant breakpoints can be happened in any region across the 3 billion genome sequence. But if the broken regions are located in the gene body and our cell tried to fix it, but it kept the form, fused the form, then we call it as a fusion gene. And following the central dogma, it can make a fusion mRNA sequence and also can form fusion protein. And if the fusion protein contribute to the tumor genesis, then we can target that as the targeted therapy development. So far, our lab contributed to these fusion gene studies in many diverse way, like fusion gene prediction, from NGS data, identification from diverse cancer types, develop fusion gene resource and functional annotation resource, and the visualization of the functional feature, deep learning model, and etc. So these days we focus on protein structure related information. So this map shows how much our fusion gene database is used in whole world. So in Fusion GDB, if you visit, then you can see all this kind of diverse annotation of individual fusion genes. So we provide all known fusion gene breakpoints. We also provide the genomic features around the breakpoint region from our developed fusion AI model. And also we provide the coding potential of individual fusion transcripts. We also provide what kind of functional features can be lost if the two genes form fusion gene. And we also provide a fusion mRNA sequence, the protein sequence. This the fusion protein sequences are imported into the UniProt database. So far, we have 100,000 to 102,000 fusion genes. So out of these 102,000 fusion genes, after checking the in-frame water frame, we have 16,000 in-frame fusion genes. So for these, considering multiple breakpoints and the multiple gene isoforms, we made 82,000 fusion transcript sequence. And then after and the running ORF finder, we have about 42,000 fusion amino acid sequence. So among these, for the manual curated ones or recurrent fusion genes, we predicted their fusion protein sequences. So total, we predicted 3.5 thousand fusion protein in this first version of Fusion PDB. And after checking the reliability, the confidence from the PLDT score and using active site residues, we this kind of criteria we selected. We chose the confident structure. So for these chosen ones, we perform the virtual screening against the known FDA-approved drugs. Anyway, so far the situation is only 1% of the fusion genes are having targeted therapy. So this time, we also know the fusion gene can make neoantigens like other types of mutations, like indel or point mutation, etc. So this time, because we have the fusion protein sequence, we thought this can be a unique information to form the neoantigen derived from fusion gene. So from this number of fusion protein sequence, after checking the unique breakpoints, total we have 13,000 unique fusion breakpoint peptides, plus minus 13 amino acids from the breakpoints. So for these number of 13,000 unique fusion breakpoint peptide sequence, we ran the typical tools to predict the neoantigen, like net MHC pen and deep HLA pen to predict the potentially interacting fusion neoantigens against the HLA-1 and 2 types. But this is a typical way, and this is the example of a BCIB1, HLA-1 interacting neoantigens, HLA-2 interacting neoantigens. And here, using our expertise, we added one more step to filter and false positives. So what we did is we predicted the 3D structure of these short peptides, and then we downloaded all known HLA molecular 3D structures from PDB, and then we, against these all fusion breakpoint peptides, we checked the interaction with these known HLA molecules. So this is showing the example interaction 3D complex between fusion neoantigen and HLA. And also we also provide the potential CAR-T therapy target in fusion gene. Since the CAR-T therapy target the cell surface proteins, we checked first how many fusion genes are formed with any of the cell surface protein partner. Then we found about 4,000 fusion genes were formed by any cell surface protein partner. And out of these, only 500 fusion genes retain the transmembrane domain. And we also, for these, and then these 500 fusion genes can make about 5,000 fusion protein sequences. So for these, we predicted the 3D structure, and also we checked the domain retention, and we also predicted the cellular localization, whether they are located in the transmembrane. So this is the example of the very famous fusion genes. So the blue these are all our predictions. And then among these, only reds are the experimentally reported fusion neoantigens so far. And our resource that we restricted to the neoantigen retained when they are crossing the breakpoints. So it is very unique neoantigen. So these are there are not many studies done for the fusion gene neoantigen. So these are all studies so far. So these are the reported fusion genes with their neoantigen study. And this is our statistics, how many fusion genes we investigated, how many fusion protein we have, and then how many HLA-1 and 2, and the 3D structure interaction, etc. So this is the top example that have the fusion neoantigen, how many samples. So BCIB1 has the largest number of samples with this fusion gene, with the fusion neoantigen based on HLA-1 and 2. And this is the 3D complex of the top 9 most frequent fusion genes, neoantigen and HLAs individuals. So this large one is the HLA molecule, and the middle one, peptide, is our fusion neoantigen. So the detailed information of individual fusion genes are shown in this table. And if you visit our website, you can download all this information for individual fusion gene cases. So predicted fusion neoantigens and the multiple sequence alignment of fusion neoantigen identified in individual breakpoints, the fusion protein sequence, the structure, and the MHC pen result, immunogenicity score, and cell surface fusion protein information, and cell localization information. So this is we want to study the pediatric sarcoma patients' fusion neoantigen cancer vaccine from this pipeline. Thank you for listening.